Hello, and welcome to the roundtable. So I'll first start presenting the different speakers that will join us today, and then we'll ask them some questions and of course at the end of the roundtable, you, the audience, will be also be able to make any questions that you want. So I will first start with Pascal. Pascal is trying to fix this. Maybe we can jump to. Are you ready to go, Pascal? Or we can jump maybe then to Isabel and Armando and Isabel. You're working on the same topic. Maybe you, Isabel, or Armando want to talk about your projects. Isabel, yes.
Yes, Isabel and Pascal are working the same use cases. I don't know if you want to start with the first one, or maybe we wait, or I start this, up to you.
You go ahead and then, yes.
Okay, so, well, this is the, in my case, in the case of ASINCAR, well, we were working in the specific use case focused on the animal sector. In this case, more specific in the meat sector. So we focus our studies in the meat quality. And yes, we collaborate in the project with ASEABA. It's a breeder association, which main task is the breeding program management of one special beef breed called Asturiana de los Valles. It's very, I would say, popular here in the north of Spain, but this type of beef breed is around all the Spain, all the country. And the use case, the objective was to study the relationship between relevant phenotypes and for the beef industry, and this this relationship with specific omic data. So we divided the study, the use case, in two different strategies. So the first one, the approach was different. So in the first one, the first strategy, we focus on the classification of carcasses according to tenderness at a specific muscle, call it longissimus dorsi. Yes, in this case, for this strategy, we collected data from more than 200 samples, specifically 222 samples obtained from male steers at the slaughterhouse, and we analyze, in terms of phenotypes, we analyze the tenderness. So we carry out the tenderness characterization of all samples, and we select the most extreme phenotypes. So we selected 10 lowest and 10 highest values in terms of tenderness. And with these 20 most extreme phenotypes, we perform as the omic analysis at different levels. So we obtain data from genomic, transcriptomic, epigenomic, and metabolomic.
Well, just in the interest of time, maybe we'll jump to the other speakers, and then we can maybe just a brief introduction of the.
Sorry.
No, it's okay. Fine. Maybe so. Now I think Pascal has the. Not working. So maybe you, Isabel, can cover him and explain a little bit your user case.
Work in the cattle fertility case with Pascal from Eliance and SERIDA. And this case has the objective of identifying biomarkers for fertility in the context of the embryo transfer technology, and also to increase the knowledge of the pathways involved in the pregnancy establishment. So for this task, we collected and analyzed plasma samples from synchronized female recipients on day 0, which is the day of the estrus, and on day 7, a few hours before the embryo transfer. And then all these plasma samples were analyzed by proteomics and metabolomics And after the embryo transfer, we collect the information about the pregnancy status of the recipient that were transferred with the embryos on day 40, day 62, and birth with the objective of identify the biomarkers for pregnancy.
Germany, and we work mainly with plant data within our group. So we are a bioinformatics group, but we work with plant data. So to be more specific, in our use case is in relation to cassava. So for this plant, it's a tuber plant, not so common in Europe, but it's certainly a very important tuber plant that provides nutrition for a lot of the world. We are interested in looking specifically at the transition of the roots. So at the beginning, it forms normal, like fibrous roots, and as the plant develops, then it develops large storage roots. So what we really want to know is what is it that actually activates the formation of these roots? And if we can understand more about this, then we might be able to pick better cultivars, which will produce larger storage roots, perhaps more storage roots. And yeah, so this should improve the yield in the long term. So that's a very brief synopsis about what my use case is.
Yes, my name is Sophie Colombier. I'm working in the same team than Pierre, who presented this morning. So at INRAE, Bordeaux, we are studying the plant metabolism. So we are a multidisciplinary team. We have some expertise in analytical chemistry, biochemistry, molecular biology, physiology, and also bioinformatics. So our objective is mainly to understand the metabolism and how the metabolism participates in the performance of plants and especially foods. So we perform systems biology with both experiments and modeling. So mainly we perform large-scale experiments, like with fruits, and our strategy consists in detailed experiment with temporal dynamics and a large diversity of species. And then we collect some information as much as possible about environmental condition and so on. And on the Bordeaux Metabolome platform, we do the metabolomics analysis for untargeted and targeted analysis. And then we combine this omics analysis with other omics like genomics and proteomics. So so then the objective is to collect these omics and then to combine and to find the biomarkers of the food performance.
Alcina?
Hello, good morning. So the team from University of Minho worked on the bioenergy production use case that focus on the anaerobic digestion process. That is a biological process in which organic waste can be converted to biogas, the mix of methane and CO2, that is a renewable energy source. And this process depends on the activity of thousands of different microbial species with different metabolic functions and distinct taxonomies, much of which is still not known. Neither is the importance of specific microorganisms or the intricate interspecies interactions that are crucial to achieve an efficient methane production. So in this project, the objective was to, by using the GLOMICAVE platform, provide a comprehensive analysis of the microbiology of the methane production by linking the microbial identity and function given by meta-omics data that are integrated in this platform to the efficiency of the anaerobic process. And in this way, achieve to identify biomarkers that are correlated with the good performance of the methane production, so that we can understand this process in a way that we can further optimize methane production from waste. So this is basically the main objective of this participation.
Thank you very much. So, Pascal, do you think that?
Is it better now? Yeah. Okay, perfect. Thanks. So I don't know, Isabel already presented our business case, so I have nothing else to add to the presentation of the business case.
How we can contribute to how Omics technologies can contribute to your use cases. For example, for you, Isabel.
So in the last 20 years, the use of embryo technologies has been increasing worldwide with now more than 1.5 million of bovine embryos transferred globally. So the main problem is that despite the technology is increasing, between the 40 and 70% of the embryo transfer do not reach birth. So this leads also to important economic losses. One of the reasons of these low pregnancy rates is the lack of efficient methods to select competent embryos and recipients. Usually the recipients are selected in base of the detection of functional or healthy corpus luteum or progesterone measurements, or simply by rectal palpation. But the problem is that these methods often fail to identify the competent recipients. So there is a need for new efficient methods for recipient selection. And as we know, in the last years, the use of omics has been also increasing. And the main advantage of the omics is that we can analyze of the molecules of a sample, including proteins, metabolites, genes. So the omics can allow us to identify biomarkers for distinguish between recipients that will be able to get pregnant or recipients that are not fertile. So with the objective also to improve the pregnancy rates.
Thank you. And around the same topic, maybe a question for you, Armando. What do you think that are the main barriers? How they can impact on the livestock sector addressed to meat production, which is your use case?
Yes, well, obviously, for sure, the main, in terms of impact, so the main impact pursued is the improvement in terms of production efficiency as well as meat sales, obviously. So this platform will provide the opportunity to improve the quality of the final product. So this is directly related with production efficiency as well as the final benefits of the breeders. So this is the main impact, but obviously omics is a technology that is not well implemented in this sector, in the animal sector, especially in the breeders, with the farmers. So talking about barriers, I will say that the lack of awareness, the existence and usefulness of the solution is a barrier. So there is probably the farmers are not informed about the benefits of this platform, that this platform can provide them, the type of information. So there is a resistance to change also, because this is a very traditional sector. And there are not many digital technologies experiences on this field. So, well, this can be another barrier. And obviously, they need the support from genetic experts in order to take them the benefits, to get benefits from this platform. So in this case, ASEABA is going to put effort in different activities in order to mitigate this, to reduce these barriers. So in terms of support to farmers through information and formation activities and campaigns for consumers, well, all these kind of things. But in the future, we hope that this platform will be implemented in the sector. Yeah.
Thank you. And let's stick to the livestock sector, but let's jump to the GLOMICAVE platform itself. Question for you, Pascal. Can you tell us what is your positive or negative experience with the Omics data in the ruminant sector, and GLOMICAVE can help you or will help you in the future?
Yes, Xavier. Thanks for the question. Regarding the omics science, our experience is quite contrasted in the ruminant sector, because in the past years we have known a very good experience with genomic analysis and DNA reading by SNP analysis or sequencing, because with that kind of omic we succeed in about 10 years to predict with a quite high efficiency the performances of animals in cattle. And so we use this application to select the best animals at the level of the whole population of the breed or at the level of the farm, to improve the efficiency, the sustainability of animal production. So it's work quite pretty well for classical and simple traits like milk quality, milk quantity, morphology of the animals. But we have some problems with complex traits or phenotypes which are difficult to measure, like reproduction, methane emissions, robustness, or everything. And for these traits, it's quite complicated to have an access to this phenotype. So we have to find biomarkers, we have to find some proxies to indirectly measure the phenotype. And here is the problem, because for many years we had a lot of publication in the scientific field for differentially expressed genes, proteins, or metabolites. So we have a lot of candidates, but finally we have no validated biomarkers, and we have some difficulties to reach this objective. And the validation step is very important for that, because the biomarkers, the proxies, need to be cheap, robust, and easy to measure at a large scale. So I think the GLOMICAVE tools that have been developed during the project will help a lot the ruminant sector to find the right biomarkers, the right candidates, and to try to get an application behind. So yes, I think GLOMICAVE platform must give us more certainty in the choice of our biomarkers we want to focus on.
Thank you. So you talk about biomarkers. That's interesting. I believe that Sophie also shares some of your problems in her use case in biomarker selection for fruit quality. So Sophie, how do you think that the GLOMICAVE platform can help you to find the biomarkers for fruit quality? How do you think that omics data can contribute to improve the knowledge on the links between the fruit genotypes and the phenotypes?
Actually, the biomarkers can help to improve the agricultural practices and fruit and plants more generally, but they represent a big economical sector, and to improve the agronomical practices can be very important, also to reduce the impact of disease like the Charka disease for plum, introduced by the viruses, the plumpox viruses. And in that case, we can expect some biomarkers in metabolomic data that can be able to detect the disease at the beginning of the infection, so to reduce the number of plant and fruit, and to limit the invasion. Also, in the case of food, we can expect some biomarkers, interesting biomarkers, to control the food growth, because some foods takes a long time to reach the maturity. So we can expect to reduce or to optimize the agricultural practices and the time to reach the maturity for food.
Thank you. So I have the same question for you, also Marie and Alcina. How do you think that the GLOMICAVE platform can accelerate data analysis and allow biologists to draw scientific conclusions? How do you think that omics science can contribute to your use cases, to biogas production, to in your case, Alcina? what do you think are the limitations, and how do you think that GLOMICAVE platform can overcome it? Perhaps you, Marie, you can start.
Yeah, sure. So I would say that from my perspective anyway, in recent years, what you can see more and more is that these omic technologies have matured a lot. So biologists are able to generate very, very large amounts of data now quite quickly, specifically, like I would say, genomics, transcriptomics data. Very often then we are left, or biologists are left with very large amounts of data that really, to analyze all this is not a trivial task. So I think the GLOMICAVE platform provides a good way to try to bring in different types of data and large amounts of data, and hopefully removes the burden of analysis to come up with, where you can potentially look at hypotheses, come up with potential targets, and then proceed with your analysis. So for me, this would be where I see a big positive with the GLOMICAVE platform.
Thank you. What about you, Alcina?
Because in the anaerobic sludge, so the organisms that thrive in anaerobic digesters, there is a complex net of microorganisms living in communities with multiple relationships among each other, and the majority is not cultivable. So understanding their function is only possible by applying culture-independent methods, and this can be accomplished by using meta-omics approaches. And of course, as we know, meta-omics approach generates a huge amount of data that is very difficult to correlate, and especially with the performance of these digesters. So the GLOMICAVE platform can provide a way to overcome this limitation by integrating all this data in a comprehensive way, so that it's possible to identify potential biomarkers, so microbes, genes, proteins, metabolites, that are linked to a good anaerobic digestion performance. And these biomarkers can then be used as targets to monitor and optimize methane production in these anaerobic digesters.
Thank you very much. Okay, so I have other questions for you. For some of you, this has been your first experience using machine learning technologies, right? Machine learning technologies applied to your use cases. Thanks to the AutoML module of the GLOMICAVE platform, you have been able to extract hidden relationships, hidden information from your data that otherwise would have required technical skills and expertise in coding, in software programming. So how do you think this has changed your view on the application of machine learning? What's your view now about this technology? I believe that you, Alcina and Sophie, have extracted pretty useful information using these machine learning technologies of the GLOMICAVE platform. Do you want to share your thoughts about this?
Yes, well, maybe I can start. Yes, this tool was very important in our study case, because, as I mentioned before, many of these organisms are not known. So even so the meta-omics information gives us unknown organisms, and we don't know much about them. So they were disregarded for a long time. And with these machine learning algorithms, it was possible to identify groups of unknown microorganisms that indeed are important to have a good efficiency of the anaerobic digestion process. So we can now focus into these groups, trying to isolate or to know more about these organisms to be able to complete our understanding of this process. So this is an important contribution.
Mm-hmm. What about your case, Sophie? Want to share your thoughts?
Yes, machine learning is very powerful to predict biomarkers, because we can find biomarkers either in metabolomics, transcriptomics, proteomics, and which is very satisfying, even in transcriptomics, which is far from the phenotype. So it's really interesting because it's easy to measure transcriptomics. And so, yeah, it's a very powerful tool. So now we have to go deeper into the variables which are behind the predictors to better understand also the biology. But it's very interesting to have these predictors yet.
Thank you. So another question that I have for any of you is that sometimes, and I think that I think there is a gap between the tools and the software developers, like myself or Maria as well, and you, the biologists or the end users, right? We provide you a tool, but sometimes you might feel that any given tool, although powerful and needed, is complex. You don't know how to start using it sometimes, or sometimes it's confusing. Our guest speakers might share this thought since they have also more experience with treating or dealing with end users. So my question is, do you have been involved in the platform? You have been involved in the project since the beginning, and we have been able to close this gap between you, the end users, and the software developers. But do you have any thoughts on how we can close this gap even further, or how to close it for new users, for new end users of the GLOMICAVE platform or any other tool? And this is a question open to any of you.
On that point, Zabi, I would say that this is probably an iterative process. I think very often, when developers, bioinformaticians develop tools, it's not always clear to them what, as an end user, that they will want to see from this data. And I think one can never get all the requirements at the beginning, or it's only when you provide a tool and people start to use it that actually, even for you as a developer, it becomes clear, oh, actually, people would like to see this. People would like to download this. People would like to, for instance, like download some of the pathways as SVGs, which I know you have implemented, but at the beginning, you might just think, okay, why would the user need to do this? So I would say that this is an iterative process, that it will take users to use the platform, provide feedback, and, constantly get improved.
Yes, any other thoughts?
Yeah, I agree with Marie, because at the beginning of the project, I was quite naive with omics data manipulation analysis, and thanks to GLOMICAVE and the platform, I'm quite more confident in it participate to demystify a bit for me the omics data approach. And so it's not perfect. I have to train myself again to use the platform, but I think Marie is true. We have to train to use the platform. It's a long process to improve both the knowledge and both the platform in the same time.
It's good to have both the vision of also all you, Maria, are both an end user and a tool developer, but you, Pascal, are just an end user. So it's good to have two different, let's say, visions. But so maybe a question for you, Sophie and Pascal, or Isabel, and also Alcina. You belong to a scarce group of researchers that integrate the, let's say, mainstream omics, right? The genomics, proteomics, or metatranscriptomics, and so on, with the more challenging, at least for me, metabolomics technologies, right? Would you be able to do your research now without metabolomics? What do you will say to researchers that currently do not consider metabolomics in their experiments?
Okay, I can start. I would say that metabolomics is, no, you can't avoid metabolomics. Is really useful, very useful, because it's so close to the phenotype, and it's a huge amount of data you can achieve. So definitely we need metabolomics.
Any thoughts as well, Armando, Alcina, Pascal? Yeah.
Yeah, I agree with Sophie. We cannot skip the metabolomic step, but with metabolomics we are more close from the phenotypes, but we have more noise around the data. So, but I definitively agree we cannot skip the metabolomic, because we have a lot of metabolites in our candidates. After GLOMICAVE, we have a lot of metabolites.
Any other thoughts?
Yeah, I would like to say also, in agreement with Pascal, that in our case, for example, you can have an animal which can be fertile in one reproductive cycle, but in the next cycle, not. So with metabolomics, you can identify these animals that will be pregnant in one specific cycle. So it's very useful.
Thank you. And a question for you, Marie, because you have a lot of experience in expression and genomics data. What do you think, what additional features do you think would boost the wide adoption of the platform by, let's say, your expression field peers?
Yeah, so I guess one of the points, okay, so just even to expand on your previous question as well about going forward, people using omics data, I would say that it's coming to the point that every biologist, every researcher needs to have some degree of bioinformatic knowledge in terms of, running tools, because this is the way forward. I think more and more, everything is going to use omics in their research. In terms of what I would say from my perspective, from the plant perspective, from the types of data that we work with, I guess even you mentioned earlier, even to get the data to the point of the GLOMICAVE platform using it, it does need to be pre-processed to some degree. Be that transcriptomics, metabolomics. So already, I guess, there's some steps that biologists will need to become familiar with. I don't know whether that would be feasible in terms of including some of this into the platform. It's a big ask for sure, I would agree, but that I would say could be something that might make it simpler, let's say, for some of the people. But I do know the challenges that come with that, because, for instance, like these are very, very large data sets. So yeah, it's just let's say, an idea thrown out there, not necessarily something that I think is feasible, but where I see that sometimes people struggle.
Yes, and I might add, not even large data sets, but also like sometimes expensive or private commercial data that you have to somehow purchase to integrate, and that makes it even more challenging. Sometimes we have the data, but sometimes we lack the economic resources to integrate that, right? It's from experience. Yeah, and related to this, also for another question for you, Alcina. So because you focus on the microbiome, which is even more challenging, right? That well, let's say different, right? Let's say, yeah, you don't have one organism, but tens, hundreds, or maybe thousands, as you said before, and some of them, as you said, are unknown, or most of them, right? So I have some questions. Well, two questions for you. The first one is, has GLOMICAVE met your expectations? But I believe it has from your previous answers. But and the second question is, what tools, in the same line as the question that I made to Marie, what tools do you or your community need, as long as they are within the boundaries of the technological boundaries that we have, as long as they can be created? What tools or features do you think we can integrate in the GLOMICAVE platform? How we, the software developers, can contribute to improve the microbiome field for your research specifically?
Well, it's exactly what you mentioned. So we work with thousands of different species, and this was being the main difficulty here to apply the GLOMICAVE platform. For us, that is a feature that would be very important for in the future development, to be able to map more complex net of different species, because the interaction between them is very important for our process. So that would be something I would like to use in the future.
Thank you. Any other
possible, of course.
What's that? Sorry.
If possible, of course.
Okay,
I see. So.
I see. Thank you. So do you have any other thoughts about this question that we have made? Any other last comments you want to add? Or anything about how we can integrate the omics technologies, how GLOMICAVE can help you in your research, or any other thoughts? Well, in this case, I thank you very much for your participation here.